Hello, everyone, and welcome back to the live stream. In the next part of our programme, I am very pleased to be introducing our Biotech Rising Stars showcase, which will be chaired by Mourad Mellal, Director at Catalent. So this is a pre-recorded session featuring pictures from up-and-coming entrepreneurs, and it really does go hand in hand with the wonderful array of innovation in technologies that we've seen so far today. So just to highlight, if you would like to make contact with the presenters that you see, you can do so using the conference app. So with that, I really hope you enjoy viewing this exciting showcase, and we can roll the video. Thank you.
Good afternoon, everyone. I am Mourad Mellal, Director of Statistics at Catalent Biologics. It is a pleasure to be here today and to chair the Biotech Rising Stars pitching session, part of one nucleus on the Helix digital event. At the core of what we do at Catalent Biologics, our mission is to develop and supply products to enhance and improve the lives of patients worldwide. Our work is powering biotech and pharma customers across the drug development cycle, from preclinical through commercial manufacturing for biologics, cell and gene therapy, biosimilars, sterile injectables, and ADCs. We have experience developing over 600 antibodies and 80 recombinant proteins, and have manufactured over 45 commercially approved products, all with the help of more than 6,000 employees across our 12 biologics development and manufacturing sites in the US and Europe. Our first Biotech Rising Star is Rob Neely from Tagomics. Rob, over to you.
Hi there, my name is Dr. Rob Neely. I'm a senior lecturer at the University of Birmingham, and I am co-founder of Tagomics. I have a company focused on epigenetic biomarker discovery for oncology in the first instance. I'm co-founder of Tagomics with Jack Kenefick. He's a PhD student, came out of my lab. In fact, I graduated from my lab. I have to let him go. He's now Dr. Jack Kenefick. He's an ex-ICUR fellow. He's a Royal Society of Edinburgh Enterprise fellow. And we're working together with StartCode, in particular Dr. Jason Mourad, who is their CEO and former CEO of Cambridge Epigenetics, and is helping to guide us through this wonderful world of biotech and how we commercialize our epigenetic platform. So epigenetic biomarkers, I'm sure you're all aware, are a really exciting field in both academia and in industry at the moment. And there's a reason for that, and that's because the epigenome is it's just like a just dynamic, responsive layer of your genome that is an excellent biomarker of disease. So if we think about DNA methylation in particular, this is probably the most studied of the epigenetic markers. It provides a second layer of information on top of our genetic code that allows us to imply somebody's age, health status, their diet, environment, whether they're stressed, and all this kind of thing. And so linking those changes in methylation patterns is really key to being able to diagnose disease at its very earliest stage using molecular assays. And that's why there's such tremendous excitement around epigenetic technologies. Now, one of the issues with reading DNA methylation in particular, your genome's huge, obviously 3 billion base pairs. You're looking for tiny methyl groups on that genome, which is just a complex problem. The paradigm, the existing gold standard, is bisulfite conversion, which takes that methylated cytosine and cytosine, converts one into uracil, which is converted to thymine by the PCR. And then we have to compare the two genomes and look for the changes and the differences in those two genomes. And that's a complex procedure. It damages the DNA molecule. And so if we have very small amounts of DNA, as you do in liquid biopsy now, these molecules can be very difficult to probe and to understand using bisulfite conversion. Hence, there's been a whole range of technologies that have been developed recently that are bisulfite free, but these are still comparative methods where we need to take multiple copies of the genome, do whole genome sequencing, and compare those sequences in order to discover new biomarkers of disease. And on the other hand, targeted approaches like antibodies I've got a long history of performing okay, but really letting people down in terms of their bias towards heavily methylated regions of the genome. So the great thing about Tagomics technology is a completely new approach using an enzyme that targets unmethylated DNA for analysis. So both enzymatic technology and chemistry for targeting the DNA, and then the fact that we're going for these unmethylated regions of the genome, those two things combined are really our unique selling point. So here's a little schematic that tells you about our technology for labeling and isolating unmethylated DNA in a nutshell. We take a genomic DNA, we fragment it, and then we treat it, we just incubate it in a pot with our unmethylated targeting enzyme, a cofactor that we synthesize in the lab, and then magnetic beads. And so we isolate unmethylated DNA on magnetic beads. We have the methylated DNA on the other hand, which we can pull out for sequencing and analysis if we want to. It's a really straightforward protocol, it's automatable, and allows us to separate the two fractions of the genome. And we think that's got really unique potential to discover novel biomarkers of disease. Here's an example of a sequencing data set from our first experiments. This is breast cancer cell line versus a healthy breast epithelial cell line and a tumor suppressor gene. We see the CpG island at the promoter for that tumor suppressor gene is unmethylated in the healthy cell line. It's methylated, so we got very few reads in the breast cancer cell line. And these kind of dynamic changes across the whole genome are what our technology allows us to probe. So we're now developing that, and our first year of operation, we're going to get to that point where we've done a clinical study on 50 or 100 patient samples. We'll show that the technology works in those cohorts, and that's our value inflection point for our next funding round in around 12 to 18 months' time. So that's us in a nutshell in a 5-minute presentation. If you're interested in the technology and you would like to know more, please do get in touch with either Jack or I, or indeed if you'd like to work with us in the future, we're really happy to have those conversations now at an early stage and start to understand your problems. So thank you for listening, and hope to see you around.
Thank you, Rob. Our next speaker is Felix Dobbs from Broken String Biosciences. Over to you, Felix.
Okay, hello everyone. First of all, thanks for the opportunity to speak here today. My name's Felix Dobbs and I'm the CEO of Broken String Biosciences, and we map interruptions in the genome to unlock novel therapies. So the introduction of new medicines requires the assessment of the damage that they can cause to our genomes. And so we're now entering into this really exciting phase of therapeutic development that is really centered around the development of novel cell and gene therapies. This has really been catalyzed by the rise of CRISPR genome editing technologies that now enable not just disease treatment, but actually targeted disease reversal directly at the genetic level. And so while these kinds of cell and gene therapies offer huge potential to be transformative for human health, there is there's a kind of double-edged sword element of this, where while CRISPR can make those targeted changes at sites in the genome that we're desired, it can also go off target and introduce mutations in places that are unintended and unknown. And so because of this, assessing the safety of these kinds of cell and gene therapies is actually much more challenging than the more traditional classes of therapeutics, such as small molecule drugs and larger molecules like proteins. And this now requires a paradigm shift towards the use of genomics technologies for this purpose. And so really taken together, new technologies combined with a data-driven approach is now required to safely translate these therapies into the clinic. And so then the problem here is that outdated and insensitive technologies impede the safe introduction of novel breakthrough therapies. And so it doesn't, for any class of therapeutic, whether it be a small molecule or a cell and gene therapy, must be assessed for its ability to introduce genome breaks prior to it being taken through to clinical trials. And this is actually because genome breaks represent a very significant form of DNA damage that is a key indicator that a therapeutic is not safe for human consumption. And this is actually because it's thought that just a single unrepaired break can lead to the development of cancer. And so according to the AstraZeneca 5Rs framework, more than 50% of preclinical project closures are actually due to safety issues, of which genomic safety represents a significant proportion. And when on average it costs around $2.6 billion to take a drug to NDA, this represents a significant problem. And so the legacy technologies used to measure genome breaks form a part of this problem, which are shown on the right-hand side. And as you can see, these are really they're outdated technologies based on imaging, and they provide a very limited readout of what's going on in the cell, and they simply lack the sensitivity to accurately assess the safety of more targeted therapeutics like CRISPR genome editing, and they can't be used to measure those off-targets. And so our solution to this then is the InduceSeq technology platform. And so if you can imagine then, within each of our cells, you have these DNA breaks, and in every different cell type, you actually get a unique profile of breaks in the genome, both just formed naturally, but also that can be induced by a given therapeutic. And so it's for this reason then, we need to have both information about how many breaks are forming, but also where they are in our genomes. We developed a novel technology called InduceSeq, which is this game-changing break sequencing technology that allows the precise digital readout of breaks in our genomes. InduceSeq produces this unbiased one-to-one representation of breaks in the genome because of these steps we've taken to remove bias and remove amplification, and we actually use the Illumina flow cell in a slightly different way to enrich for our break ends. And so in the third column here, you can see the difference between our InduceSeq readouts versus alternative approaches where, because they've introduced these kind of PCR biases and in several steps of the process, you get this simultaneous gain and loss of signal, which really makes these approaches difficult, the data produced by these approaches difficult to interpret and makes it very difficult to be actionable from this data. And so we're now applying our InduceSeq technology to the application of measuring off-target effects of genome editing, because we can produce this digital readout of CRISPR off-targets shown on the right-hand side. And what makes our technology different here is that when we're measuring these sites, we now can take a quantitative readout of all of the different off-targets and compare that to the on-target site. And this is something you simply can't do when you distort your signal in such a way. And so really taken together, Broken String is building a platform that will enable a data-driven approach to the development of novel therapies that puts patient safety at the core of the design of the therapy right from the start. And so what's really cool about our technology is, although it can be used for this kind of genome editing off-target assessment, there are also a number of other therapeutic sectors that can be addressed using our technology. So there are genome breaks, because they're quite fundamental, they're involved in many different areas. And we believe our technology can be really bring a lot of value to doing things like developing novel cancer therapies and even better understanding neurodegenerative disease. So we are now part of one of the cohort companies of this year's Illumina Accelerator program, and we're working with partners to translate multiple proof of concept projects into long-term co-development partnerships. We're working with a lot of different exciting partners in some different areas, including the UK Regenerative Medicine Platform, Horizon Discovery, and AstraZeneca. So just very quickly, acknowledgement of our founding team. There are 4 co-founders of Broken String, including myself, and that's Professor Simon Reed at Cardiff University and Dr. Patrick Van Eyck, as well as Simon Kerr, who joined us in 2019 to set up the company and spin out. We're also extremely lucky to have a really excellent scientific advisory team, including several academics and on the industry side too. So thank you for listening, and I'll take any questions.
Thanks a lot, Felix. We'll now hear from Laura Towart from Vivan Therapeutics. Laura, the floor is yours.
Hi, I'm Laura Towart, founder and CEO of Vivan Therapeutics. Up to 75% of first-line cancer treatments fail. Current cancer treatment focuses on what's similar among patients by identifying single mutations and then drugging that target without addressing the complexity of real-world tumors. Advanced flight simulators allow critical decision-making to be learned safely. Our cancer simulator for oncologists can eliminate dangerous trial and error therapies on patients, and it also powers biopharma. Our technology, the personal discovery process, generates novel, personalized treatment recommendations. This platform was developed and fully validated at the Icahn School of Medicine at Mount Sinai Medical Center. The personal discovery process begins with whole exome sequencing of a patient's tumor and blood, identification of mutations or alterations leading tumor genesis, and incorporation of up to 20 of these mutations into fruit flies, creating avatars. Up to 500,000 fruit flies are grown for each patient. Then we screen all FDA or EMA-approved drugs alone and in combination to find novel combinations that target the patient's tumor specifically. Each effective combination can then be prescribed by the patient's own oncologist. We've had success in the hardest to treat patients, even after multiple other treatments failed. Mount Sinai initiated an N-of-1 clinical trial in 2015. Initial results have been very positive. For example, one patient, a 53-year-old with KRAS-positive colorectal cancer, had tried every other treatment available to him without success. Sequencing analysis identified 9 genetic alterations that were modeled in the fly, resulting in the identification of a novel combination. After taking his personalized drug treatment regimen, he had a 45% reduction in tumor size in 27 weeks. He also reported an improved quality of life. Our scalable in vivo model allows us to do what no other model can. We create a biological twin of the patient, which replicates the full tumor complexity in a living biological system and rapidly tests thousands of drugs and combinations. And then treatment recommendations incorporate non-cancer drugs, making them less toxic and more affordable. We harness the complexity of biology to give pharma and oncologists the most powerful decision tool. Patients with similar tumor genomic profiles respond to the same treatments, so our unique data set trains an intelligent platform to get better, faster recommendations in the future and at lower cost. We pair omics data and our own proprietary high-throughput fly screening data with deep machine learning processes to power a virtual and experimental lab space for pharma and to power decision-making for oncologists. Our platform technology can be used for drug discovery, optimization, repurposing, or differentiation. I'm a scientist turned biotech entrepreneur. I'm also the co-founder of Cellmatics, a company that pioneered predictive analytics and genomics for female infertility and women's health, now prospering for over a decade. I'm supported by an experienced board of directors with leaders in medtech and personalized medicine. Our scientific team is led by Dr. Nawal Villegas, who has years of experience building fly cancer models and performing drug screening. We've assembled an outstanding team of experts in cancer biology, genomics, bioinformatics, and analytics. Our medical advisory board is comprised of leaders in oncology and innovation, including Professor Ross Kagan, the inventor of our platform technology. We are partnered with oncologists at over 15 medical centers, each treating hundreds of patients annually. Our initial market is private pay, but will expand to all patients once our digital therapeutic is validated and reimbursed. We're looking for partners to help us in realizing our vision. We're raising $7 million in a Series A funding to enable us to scale operations and our commercial organization to fund the completion and validation of our digital health tool for colorectal cancer. We are revenue generating from both pharma and patients, and we have projected revenues of over $100 million by 2026. Our ambitions begin with cancer, but we can utilize our fly-based screening platform for all diseases with a genetic cause. Please support us in personalizing healthcare and stop experimenting on patients. Thank you.
That's great, Laura. Thank you very much. Our next Biotech Rising Star is Leo Wossnig from Raku. Leo, over to you.
We are Rahko, and we are building the world's first quantum drug discovery pipeline. Our goal is to address a current need to develop novel small molecule drugs that can address a range of conditions that are not adequately treated by existing drugs. Our focus is on cancer and inflammation drugs, and we aim to address novel mechanisms to overcome current challenges with drug resistances as well as with hard or undruggable targets. These are, for example, intrinsically disordered proteins or protein-protein interactions. For these areas, existing computational approaches had little success due to either the lack of speed or accuracy. On the one hand, we need the speed in order to be able to screen through large chemical compound spaces. In fact, only then, if we screen sufficiently many compounds, we are really maximizing our chances of identifying a good and promising lead or hit candidate, even for challenging targets. On the other hand, we need to also have a high level of accuracy of the predictions. This is necessary since we otherwise would be unable to reliably assess each candidate, or on the other hand, even create good hits in the first place. Rahko is overcoming these challenges with a unique combination of three key technologies. These are computational chemistry, quantum machine learning, which is a specialized form of AI, and quantum computing. Through a combination of computational chemistry and quantum machine learning, we are already today orders of magnitude faster and even more accurate compared to industry standards. And using the power of quantum computers, we will in the future be able to attain accuracies that are entirely out of reach for classical computers today. So by combining these technologies, we are able to address both the speed and accuracy bottlenecks in traditional approaches. So what do we do with our technology? Raku works in the early phases of drug discovery, in particular on identifying good hit candidates and turning these then into good leads. So we engage with our partners to help them identifying hit candidates and really optimizing them to obtain the best possible lead compounds. And we use a range of methods which are here summarized in a slide. In the hit identification step, we start with given targets, for example, the crystal structure of the protein and the binding site. We then start by generating drug-like compounds, meaning molecules, that are likely to be good hits. What you see here on the left hand is a benchmark of our generative models on the guacamole benchmark, which is by Benevolent AI, where we have indeed achieved state-of-the-art. Combining these generative models with methods that can reliably predict the binding affinity, we are then able to generate compounds that are also good hit candidates. This works since we use state-of-the-art predictive models for predicting binding affinities and activity. And here on the right-hand side, you see, for example, the performance of our method, which is the best in class, again, on PDB bind. Indeed, we have also shown that we can conditionally generate drug-like molecules that have a high activity against the desired target. Here, for example, EGFR. We have verified this, in this case, by comparing the generated molecules to the closest similar compounds that we found in Campbell. And indeed, as you can see, these are remarkably similar. In the hit-to-lead phase, we are leveraging a range of ab initio methods, for example, to perform enhanced molecular dynamics, or also docking simulations on the fly and use scoring functions which are really based on ab initio calculations. This allows us to identify the best compounds, again, from the identified hit compounds and take these forward to the lead optimization stage. In the lead optimization stage itself, we use our in-house developed methods for admin tox predictions, meaning pharmacokinetics. In fact, we have demonstrated with a large pharma client that we achieve state-of-the-art predictions for many endpoints based on our quantum descriptors. Our sweet spot is, however, the hit identification stage, and here's where our methods have achieved the largest improvements. Indeed, we have demonstrated speed-ups and cost reduction of up to 100 times compared to industry standards, which allow us to screen drastically more candidates and therefore really help our customers to identify the best hit compounds and then take these forwards. We do all of this with our quantum drug discovery platform, Hyrax, and we have indeed demonstrated the performance on multiple stages across multiple customer engagements, blind tests, and industry benchmarks. We are currently seeking additional drug discovery partnerships where we can provide our expertise for computational design of novel hits and help bring these then to the clinical stages. So if this sounds interesting to you, please feel free to get in touch with us. You can find my email address here on the slides.
Leo, thank you very much. We'll now hear from Steve Allen from Depixus. Over to you, Steve.
Good afternoon, and thank you for the opportunity to talk to you today. My name's Steve Allen, and I'm Chief Business Officer at Depixus. We're a company based in Paris and Cambridge, and we're developing tools to unlock the secrets of the dynamic genome. So let me start by explaining what I mean by the dynamic genome and why it's important. Over the past 20 years or so, scientists have built an enormous wealth of knowledge about the 4-base DNA code of the static genome. For every living organism, it is fixed at fertilization, and it's been described as a basic instruction manual for life. However, it cannot explain how a single cell with one genome can create thousands of different cells, or indeed how diseases are caused. The secrets to these lies beyond the static genome in the dynamic genome. The epigenome, with the modified versions of the 4 bases which determine the functions of the cells, and 20 of these modifications are known to exist to date. The transcriptome, or the world of RNA, which drives and regulates protein production. RNA is also heavily modified. Around 170 modifications have been identified to date. And finally, the interactome, interactions between DNA or RNA and protein or drug molecules. Unfortunately, although we've now got excellent tools to decipher the static genome, there are no robust or comprehensive tools to decode the dynamic genome. Most rely on repurposed DNA sequencing tools, which have serious performance limitations. And as a result, the dynamic genome remains poorly understood. And this is the domain Depixus aims to unlock with our Magna technology. We're developing an instrument and consumable system that can interrogate up to millions of single molecules of DNA and RNA, and not only identify the sequence, but also directly detect base modifications, as well as the kinetics of binding to other molecules, all in the same sample in the same experiment. And here's how it works. The underlying technology is deceptively simple. It's effectively a highly accurate molecular scale measuring tape. So first, DNA or RNA molecules are converted to hairpin structures, with one end bound to the floor of a flow cell and the other to a paramagnetic bead. A magnet attracts the bead, opening the hairpin, and interrogating molecules attach themselves to the open hairpin. As the magnet is retracted, the hairpin closes, but with transient pauses as the attached molecules block the hairpin. An optical measurement system tracks the position of these blocking molecules to nanometer or single base precision. For a video on how Magna works, you can use the link in the corner of this slide. So here's an example of how Magna can be used in microRNA analysis. LET7 is a family of microRNAs with 7 sequence isoforms. It's known to be a strong suppressor of tumor progression in lung and breast cancer. Base modifications M6A and M7G affect their structure and function, so drug targets require a detailed understanding of both their isoforms and their modifications. So to set up for their analysis, we first generate bait hairpins to capture the native isoforms from total extracted RNA. Then a series of oligo probes are used to identify or fingerprint the different isoforms in the Magna flow cell. Finally, antibodies are flowed into the system to detect the base modifications. This is an example of a simple and rapid microRNA identification assay that can be used as a panel test. The same basic approach allows Magna to address a variety of applications across the dynamic genome. For example, identifying DNA epigenetic biomarkers for liquid biopsy, looking at RNA-based modifications for viral pathogenesis as disease biomarkers themselves or as drug targets, and measuring the dynamics of drug nucleic acid interactions for drug development. So, to summarize, the world of dynamic genomics is an exciting and growing field that is underserved by current technologies. Depixus Magna is a new and different approach which promises a breakthrough in dynamic genome analysis, with applications in research, clinical diagnostics, and drug development. Many thanks for your attention. If you'd like to learn more about Magna or collaborate with our team, please get in touch at info@depixus.com.
Thank you, Steve. A great pitch. Our final Biotech Rising Star is Bakul Gupta from ImmTune Therapies. Over to you, Bakul.
Hi everyone, I'm Bakul Gupta, one of the founders and CEO of ImmTune Therapies, where we're designing off-the-shelf solutions for in-body cell therapy. Now, CAR T therapies are an example of cell therapy where we harness the power of our own immune system to treat diseases such as cancer. The 5 approved CAR T products have recently been providing miracle cures to previously untreatable blood cancer patients with extremely high remission rates of close to 65%. However, these powerful treatments are only available to a tiny proportion of patients across the globe. And the main reason why this is the case is the way these therapies are currently manufactured in an external biomanufacturing facility. This external or ex vivo manufacturing of these CAR T therapies has led to 3 key limitations which has prevented the upscaling of these therapies. So the first limitation being the really long time to treatments. So currently here in the UK, patients once approved for CAR T therapy have to wait anywhere between 3 to 6 weeks to receive their CAR T cells, which reduces the eligibility of these patients by 70%. The second key limitations currently faced by these therapies is the exorbitant cost of these therapies. So again, NHS right now pays over £300,000 per patient per dose for the autologous CAR T therapies. And finally, the last limitation that I will talk to you today about includes the unreliable potency of the CAR T cells so produced in an external manufacturing setting. So to overcome these challenges, what we've done at ImmTune is to eliminate the need for this external manufacturing, and instead we've given the power back to the patient's bodies. So we have developed an injectable formulation called the nanocard, which consists of delivery vectors which would deliver the genetic payload, in this case a CAR transgene, to very specific immune cells inside the patient's body. So the way this therapy works is once we inject the nanocard formulation via an intravenous administration into the patient, the delivery vectors very specifically find and target specific T cells inside the patient's body, deliver the CAR transgene into these T cells, and then these T cells are engineered within the patient's body to produce CAR T cells, and then these CAR T cells expand and activate inside the patient's body to clear out the patient's cancer. Now, the key benefits of nanocard or doing in vivo or in-body CAR T therapy leads to 3 key advantages of increased potency, because we're using the body's innate environment to create these therapeutic CAR T cells, a faster or instant dosing, so the patient does not have to wait for 3 to 6 weeks for them to be reinfused with their CAR T cells, and overall an affordable treatment, overall reduction in the cost of these therapies. Now, these 3 key benefits of nanocard would ultimately lead to an increased patient access in the future. So we incorporated ImmTune in the summer of 2019, and since then we filed our first patent. We've raised investment from both US and UK based investors, expanded our team, and are developing our first prototype as we speak. And along with that, we are very much interested in partnering with academic and other biotech companies active in this space. We are also currently raising a seed round, which would help us to validate a technology completely in vivo in the relevant animal models, and also produce a prototype fit for human use right before we apply for our IND application. So we are a team of experienced scientists, biotech professionals, and entrepreneurs who are aligned in the vision of making cell therapies and delivering these cell therapies to all the patients in need and not just a few. So if you're interested in learning more about our technology, please feel free to contact me via our website, www.imtunetherapies.com, or via my LinkedIn page. Thank you.
So thank you for your participation. If you have any question for myself or other speakers, please come back to us using Vivan app.